Good morning, everyone. I'm really honored to present my work today. Since 2012, a global decrease of the use of antibiotics has been observed in Belgium, probably due to new recommendations on their use in livestock. Moreover, since 2016, a new regulation on their use is applied. For instance, antibiotics classified as criticals in human medicine may not be used without a prior sensitivity test showing their activity and the absence of activity of other antibiotics on the bacteria responsible of the infection. As a consequence, the Regional Animal Health and Identification Association, or ARCIA, have recently observed a decrease of resistance to third and fourth generation cephalosporin, classified as criticals, in bovine E. coli. However, the actual evolution of each family of β-lactamase-encoding gene is unknown. Therefore, the purpose of this study is to detect which particular blood genes and blood enzymes are concerned by this decrease. Isolates used in this study were collected for 4 months following the diagnostic routine of ARCIA. To make a long story short, the E. coli were isolated from fecal samples or from intestinal contents and organs of necropsied calves inoculated onto Gassner agar plates. From fecal materials and intestinal contents, 3 coliform isolates were subbed on 3 agar plates. Minca to perform agglutination tests to detect Fimbriae F5 and F17A and the antigen CS31A. The enterohemolytic agar plates to detect enterohemolytic isolates. When one positive isolate was found, the disc diffusion assay was performed. When the culture of organ was pure, the disc diffusion assay was also performed. If the sensitivity tests show resistance to beta-lactam antibiotics, the isolates was collected. In parallel, we used the McConkey cefotaxime agar plates to detect the production of a cephalosporinase, an extended spectrum beta-lactamase, or a carbapenemase. Growing isolates were also stored. For the disc diffusion assay, we used these following 8 beta-lactam, so they come from various class: aminopenicillin, different generation cephalosporin, and carbapenem. We also use beta-lactamase inhibitor. The actual classification of beta-lactamase genes and enzymes is complex, not to say very complex. It's why we use this simplified classification in this study. It's based on the phenotype observed at the disc diffusion assay. So we have the classical BLA with resistance to aminopenicillin and sometimes to aminopenicillin in combination with beta-lactamase inhibitor. Classical BLA associated with a resistance to cefoxitin, a second generation cephalosporin. USBL with resistance to cephalosporin. OMC, which are resistant to cephalosporin but also to the inhibitor of beta-lactamase, and the carbapenem, which are resistant to all category of beta-lactam. Of course, we hope not to find any of these in our collected isolates. After the getting of whole result, it was decided to test this following 94 isolates with this resistance profile and these virulotypes. So we choose 33 classical BLA, 17 of them with resistance to cefoxitin. A majority of USBL isolates, 50, 17 of them showing a combination with a classical BLA with or without cefoxitin resistance, and 11 OMC, 17 of them with intermediate and non-full resistance level. So for the microarrays, we do the DNA extraction with the blood and tissue kit from Guerin, and we froze the DNA until the microarray test. We use the Czech MDR CT103XL to perform the microarrays, and the advantage of this test is the capacity to detect SNP, so the single nucleotide polymorphism. This test detects many blade genes family, Like that, we can find TEM wild type, which consists in TEM1 and TEM2, not differentiable. Some USBL genes, like 4 groups of CTXM and 4 subgroups for CTXM1. They contain many genes. And different variants of TEM and ISHV USBL, which differ by only 1 SNP. 7 OMSI genes, and of course, some carbapinimase genes. In all bovine isolates, we detect these following genes. So we have 23% of isolates at TEM wild type only. The USBL genes were detected in 53% of all isolates, and they come only from CTXM groups. And the OMSI genes were detected in 6% of the isolates, and it consists in SEMI2 and DHA. Because many isolates present 2 or 3 genes, the TEM wild type was the most prevalent detected gene. However, 18% of our isolates was negative at the microarray. So in summary, we detect TEM wild type coding for classical BLA, different groups and subgroups of CTXM coding for an USBL, 2 genes coding for an OMSI, and hopefully we don't find any carbapinimase gene. We have compared the phenotype observed at the disk diffusion assay with the detected gene by microarray and found 3 categories. The CAT1 isolate with a perfect match. The gene perfectly explained the phenotype. CAT2, the isolate with a partial match. So the gene can explain the phenotype only if we hypothesize co-expression between genes. For example, if we have a CTXM gene and a TEM wild type gene and the co-expression, it can explain the resistance to amoxicillin and clavulanic acid, which usually is not observed in USBL phenotype. And CAT3, the isolate with no match. So either we did not detect the gene or we detect the gene with another phenotype observed. For example, we detect the BLAST CTXM genes coding for an USBL in an isolate which present OMSI phenotype. We did not find any relationship between the virulotype and the detected genes. It looks as if the majority CS31A and negative are USBL positive. Therefore, these results are biased since there is a majority of CS31A in our collection, and since we choose a majority of USBL positive isolates to perform the test. When no genes were detected in the isolate, there can be different explanations. Firstly, we have to keep in mind that the microarray targets a limited number of genes. Therefore, we can have other blood genes or another variant of the gene we are trying to detect. A second possibility is to have a different mechanism of resistance. Indeed, the resistance to beta-lactam can be mediated by efflux pump, modification of the PBP, or a change in the permeability of cell membranes. To investigate this hypothesis, different techniques can be used, as the whole genome sequencing, the PCR, or the specific phenotypic assay. What can we remember from this study? The high proportion of USBL in calves is due to the selection during the collect and to the choice of isolates to perform microarray. But on the USBL detecting gene, they come only from the CTXM groups. For the majority of the isolates, there is a concordance between tests. And for undetected gene or mismatch, other analyses are necessary. So thank you very much for your attention. ご清聴ありがとうございました。